In order to start this roundtable, I would like to invite to coordination Mr. Helvécio, Vinicius, and Rocha, and ask our speakers to come back to the table for debates. Good afternoon to all. I would like to thank Dr. William Wiseman for the invitation to participate in this table and would like to congratulate speakers for the excellent speeches. Just a remark. I would like to. I would love to ask the first question. A remark we've seen on presentations is that initial results are more focused on. That's my impression, at least, in the diagnosis part. I can understand that we have more diagnosis than treatment as a coincidence by presentations, or is there any subliminal message that we will have diagnosis available more faster? And in the chain of values of the pharmaceutical industry, new chemical entities are the ones that have more added value in the world. And we know that the world pharmaceutical market, is suffering a problem because these products are less launched in the market. It's not only a Brazilian problem, it's a world problem. Nanotechnology can fill out this place using some technologies and some synthesis routes, talking of new entities, and trying to bring this innovation that is so needy in terms of pharmaceutical development. This premise may seem interesting on the first moment, but after presentations, there is a doubt. If the next barrier will be the difficulty in the synthesis of new molecules, or will it be a nanotoxicology? And will this nanotoxicology appear by studies or by the lack of regulatory framework when they come to the patients? I think anybody can answer the question. It's not directed to anyone specific. Since I only remember the first question, that's a lot of information. I'll try to answer the first part of it about biosensors and therapy, right? I think both fronts are very important in nanomedicine. These three fronts, regenerative, diagnostic, and therapy, must walk together because otherwise it won't work. Of course, there is an advantage, an experimental issue there of experimental facility that needs to be considered. It's much easier to produce sensors, biosensors, trying to detect anomalies on the standpoint of ethics or committee material for therapy, where you depend on special facilities, on real samples of donors, of outpatients, clinics, ethic committees. All this may take longer. The biosensor is much faster than to be put in practice because you don't need anything of these. You can work with standard or pattern samples without needing real samples to prove your concept. So this experimental facility of biosensors has happened faster because there are research institutions that have no link, no interface with the biological or medical part, which is as fast as others. So it's more difficult to get some therapeutic work. So this may be an excuse for giving, due to the advance of this biosensors sector. I've seen that nanomaterials for therapy have advanced in a very terrific way. It's very gratifying as detecting and eliminating these diseases. I think both are walking together very well.
Okay, you remembered the first question. I remembered the second. So there was
Wonderful.
Okay, so we complement each the other. Basically, there was a question or comment regarding that most of the studies are most focused on the diagnosis. Basically, this is right. For a proof of concept with nanosensors or with nanotechnology, it's much easier to get a proof of concept with diagnosis because it's the population is much more discriminated. Even if the reference or the clinical conventional instrumentation which can be used to our purposes are already exist. Nevertheless, the more interesting data are found in the screening. Basically, the screening is to take a population which we don't know whether they are healthy or not. Basically, these are healthy people. They are considered healthy people, and we want to see whether they are at high risk. So basically, more and more, once we start our point in the diagnosis, we can continue with the screening. But in the case of the screening, we need a large populations and, much more important, a large amount of nanomaterials. And more importantly, for the screening, we need to adapt the nanomaterial in very fine-tuning manner so we can target the screening. So I think this is our main target, and the speaker have also focused on it. We more and more people, more and more scientists in the field are going to the screening because we believe this will be the real advantage of the nanotechnology. And of course, again, the ability to change the size and to tailor the size with the optical properties, mechanical properties, as well as with the chemical properties will be a huge advantage for the screening as well as for the prevention. Further to this comment, the targeted therapy, it's an important thing to do, but without diagnosis and screening, good screening, we cannot do targeted therapy. So I think there are many fields here in nanotechnology which are complement each the other, and part of these parts were also targeted by the second speaker, and which was nicely, but again, we are still at the beginning, and we need to explore much more nanomolecules or nanomaterials in order to achieve high sensitivity and specificity values.
I just would like to make a comment about this last question. There is a neglected part of it we are trying to underline, especially in the health area, which are the correlations of nanomedicine and nanoveterinary. In veterinary, when we talk to vets, that most of the active principles of molecules launched in the past were abandoned because of effectiveness. Sometimes they're abandoned not because of effectiveness of the active principle related to the disease, but because of the interference that happened during the action process. I believe things are a little bit parallel. We're still looking for new molecules because we need more effectiveness to tackle other diseases. But when we have other application strategies, that doesn't mean that we look for valuing those molecules that lost their patent right. In developing countries like Brazil, it's it helps to develop some kinds of medicines that are of public domain and that are approved but need to be worked with high doses, only with extremely innovative technologies that make a new price in the market and a new input. It's important to remind that the technology and the protection associated to the vehicle is not always associated to the molecule. So, especially for vets, I can talk about vets because we're more interested in them. But especially vets are becoming more real. I think we have two or three uh, products which are old molecules that are protected and last longer. For those who deal with the milk, cow milk, for instance, it's interesting to have more time to remove the same quantity of milk for a longer time. Only the application strategy has changed, so we can look for another valuing alternative. Thank you. Now we would like to give the floor to the public to see if anybody would like to ask a question. Please. I would like to congratulate all speakers. My name is Ana Luisa. I'm dermatologist of the occupational dermatosis at Fiocruz here in Rio. My question is to Kawe. You mentioned the coating of objects with nanoparticles, with bactericide nanoparticles. How is the bacterian resistance? And another question about the bacterian flora and digestive tubes in the skin. Don't see it as only pathogenic bacteria, or are you creating new diseases by using tissues or common instruments? That's a very good question. 2 years ago, we had a contact with the hospital in Jou. They said if we have a bactericide system with constant contact with the human body or another biological means, you can favor a more resistant bacteria. The infection would be more serious for the individual, but there is a certain resistance to use bactericide systems where they are in the non-continuous, like pinches or gloves. These are realities. They already have sterilization processes, and the bactericide can enhance the safety of sterilization process. The main focus when this get mixed with technology, the main focus of bactericide systems is associated to those systems that have contact with many people in very long terms without a general control. For instance, general use equipment in hospitals. I'm not talking of contact, I'm talking about chairs, like dentist chairs, seats, chairs, or systems that have low level of control. Within these systems, we can, to improve the asepsy level, you're reducing the chance of preserving existing bacteria for other sterilization systems. In fact, this needs to be better studied and analyzed, but it's good to remind that some bactericides in the market are very well accepted, like triclosan, for instance. It is complicated because you use it for carpets. Even the light can be enough to make it evaporate and generate new occupational problems. In this situation, for instance, a silver particle can be a candidate for triclosan by volatile contamination. I insist that the application of the property observed has many applications, but the application has its specs. You have to watch them And you're right, the continuous contact system, we need to be careful when we talk about it. 3 questions. When you show there is a diagnosis of leishmaniosis with antigen, don't you think is it causing the same false positive result than the same we would have in the other way? That would be more an advantage. May I pass to the second one? Okay. The second one is I have no knowledge about it. So every nanocomposal, when you work in bioassays, becomes not doesn't become a nanocomposal when it's recognized by cells, as you showed in your in vitro models with cellular cultures. And the third one is linked to the in vivo studies. Which are the parameters? Which are the analyses made for chronic assays for toxicology that is more chronic? Now I remember the 3. Okay, the first part is the answer is no. That system of biosensing standard biosensing system, like a biosensor, we always have a bioreceptive platform that will report us by means of an optical or electric sign, like a molecule. In our case, this platform is the antigen. It's an immunoassay. We're talking about antibodies. We have antigens of leishmaniosis. In the system of conventional assays, such ELISA, for instance, the same antigens from leishmaniosis can act with anti-Chagas and anti-leishmaniosis systems. We have the optical sign. When this reaction occurs, there is a peroxidase, which gives an optic illusion. In this case, I can have in my biosensor the antigens against leishmaniosis, but the electrical measures is different from one and the other because both occur with different intensities. So I'm using this electric detection strategy to differentiate. I have to differentiate it between the two events. The second question was about right aggregates. You mean? This is very clusters. When we make in vitro studies, we put nanoparticles in solution, a tampon, for instance, in the same tampon, in the culture means. But in this culture, there are proteins, there are albumines, there are everything you can imagine. We have to take care before putting this culture in cells. We have to make a new analysis of nanoparticles because you're absolutely right. There are nanoparticles with positive load that get complex with many ions, and other ones get complex with albumines. They become very big. We have to take care and identify what happens with nanoparticle when it's after entering the culture, before entering in touch with the cell. When aggregates happen, we use another system in order not to have aggregates, but the inversion of load can happen any time. We control it. It's interacting with the cell with the positive load, not anymore with the negative load. So I have this control. If I interact with proteins such as albumine and form an aggregate, it's not what I want to study anymore, so I have to discard it. I have to do the analysis before. So that's an important point which has been discovered lately, very recently. I was in a medicine congress in the United States, and they have discussed this subject very much. So when it interacts with the cell, I have to know which is its size and its load. That's easy to detect. And the third one was about the monitoring in vivo. Okay, in vivo we only used rat models. Have you evaluated 30 days, 90 days? What did you do? No, these assays were of 20 days. I have to double check these figures in some place. But more than 15 days, but it didn't come to 60 days. I don't know it by memory, but I have all parameters. We can talk about it later. I can give you these details, okay? Thank you. My name is Claudio Tedesco. I would like to congratulate the table. We're only asking a few questions, answering a few questions. The fact of looking for new molecules was a reality that has changed. We cannot forget that until we produce a molecule and it comes to the market as a drug or a medicine, we're talking about 10 to 15 years. So pharmaceutical industry has remodeled its way of thinking and came back to the health sector, human health sector, looking for old molecules with new coatings. This is how we get a new aspect. These molecules that were not used anymore because of side effects or dependence are becoming stable and creating a new profile. This is the reality that has moved not only the pharma industry but all the research related to the application of these molecules for health, especially because they bring with them a history, a clinical history that is important and makes it advance from phase 1 to 2 and to 3. Regarding application and diagnosis, this is also applied in health. The problem is that we have a regulating framework for the production and development. The application in human health is highly regulated until we get all the approvals in the research and ethics committees and human committees. This has been happening in parallel. Thank you. I just would like to compliment that I agree with the issue that how nanotechnology can bring old new products to the market. The question, the number of registrations of new molecules is dropping every year. This is the real fact. One doubt, I'm encouraging everybody to analyze this depending on the technology that is used for a nano area. If you make a registration at Anvisa with a common product, there will be no differentiation of criteria. Once you depend on specs, the Anvisa technician will be able to detect this difference. Because some technologies are quite simple. If this is not characterized as a new product and doesn't undergo clinical and preclinical studies, and not all specs of such as toxicity will be detected, we will have a registration of a product that will be available for the population. And when it comes to phase 4 and is used by the population, adverse events that are related to nanotechnology will arise. This is one doubt I have. I hope I was clear. My name is Tanya. I come from the Federal University of Sergipe. I'm a sociologist. I work at the Sociona network. You must ask yourselves what a sociologist is doing here. I would ask Dr. Rocha, what drove my attention in your speech is that it was the first time I saw this worry that the product has to be easy to use, has a simple analysis, fast in the results, and has to be cheap. This word cheap drove my attention to public health policy. I live in a country which is not a developed country. It is a developing country. That's the reality. That's the truth. Health areas are very needy, especially public health. When we have some advanced tools to detect a disease, it is extremely complicated to give access to to most people. So I became happy when you said it was easy. So that's terrific that you are concerned about something that needs to be democratized because the research is financed with public money. People pay for research that they don't use when according to what is produced. So I ask you, Dr. Hossan, this machine of measuring machine, is it already produced? Is it used? And if yes, is it cheap? Is it uh, available for the population in general, or is it just a selective medicine? Only those who have money have access to high technology. Those who don't have to look for the healer.
Thank you for the comment and the nice ideas and nice opinion. Basically, today more than 15 centers have this device worldwide, but before achieving the FDA approval, we cannot go to the commercial level or the technological level. Basically, there are many centers in Europe, in Israel, in the United States, in China. I hope next year we'll hear about the device in Brazil and other places. So we can basically right now concentrate on the research level of it. Before 1 month, the Nanos company was established, and we will start applying for the FDA. As for the cost, indeed, today in our labs we can produce this device more or less by $5,000. The cost of each sensor is more or less $1. You know, it's produced in our lab by $1. So if we go to a commercial or technological level, I think it will go down to few cents only. So it will be available not only for developed countries, but rather for developing countries. And this is the main idea, that this device should be used for screening. I would love to give one example. In the case, for example, in cancer, nobody can feel the effects of the cancer till the advanced stages, where the survival rate is very, very small, which in the case of the lung cancer is 15%. And if we target that this device will be applicable in each clinical institute or a physician clinic or family clinics or at home, then we can do the screening for everybody, both in the developed and developing countries. In the developing countries, I see this device much more important than the developed countries. In the developed countries, in the case of the diagnosis, there are a lot of alternatives, not with the same quality, but there are alternatives. In the developing countries where we have villages, remote villages, where nobody will be there or nobody can go there easily, this device will be quite good start, just to direct the people to the town in order to get further diagnosis or target therapy. So this is more or less our intention, and this is why all of these features were put inside one device, and indeed most of them we have right now in one package, and hopefully within 1.5 years we will be in the FDA and we will see it soon for everybody. Thank you.
Dr. William, por favor.
Just coming back to the aspect regarding the cost. Many of old drugs and the new presentation, nano presentation. What has this represented in terms of costs? One doses of therapeutic that costed $30 when it's liposome, it can cost thousands of dollars. There's no doubt that is much less toxicity. In that case, it's much better, and maybe our issue is that in Brazil, where we have a single universal health system where everybody has right on, that the public laboratories or the government should invest in this, because obviously it's impossible to the population to buy this instrument. We have a good example. We have difficulties to find noliposomol, and there are many other examples that are difficult to find. Even if we find a medicine with a nanostructure, the value of the market will increase highly because of the patents in our country. And aiming to the unified system of health, the investment of the government would be more than needed. This would be almost, let's say, mandatory constitutionally. Unfortunately, we don't have any time. If you have more questions, you can ask them directly to the speakers. Thank you for the insertions, and thank you for the speakers.